Hello, I'm Scott. You're interested in soil DNA testing. I'm here to concisely and thoroughly tell you what it is you will get if you submit a BeCrop sample. So, soil, as you know, it's got physical, chemical, biological characteristics. This is a test that's going to assess soil biology. It is the neglected factor in agronomic management because we have not had a good means of measuring it and therefore a good means of managing it, seeing what our management is doing to our soil workforce. The physical is the workplace, the chemical is the inventory, the goods, the resources, and the biological is the workforce, the ones who are able to transform that workplace, to move that inventory, those resources into a crop available form or out of crop available form. Is biology important on your particular operation? I guarantee you it is. This is beach sand. This is beach sand taken in mid-February in Boston. It was cold, nil organic matter, salty, and you can see this grain of sand is covered with life. I guarantee every single square inch of your operation is lousy with a soil workforce. They could be sitting on their hands, could be working for you, could be working against you. There's no way to know without taking a census and getting them on side. The more work they do for you and for your crop, the less that you, your machinery, and your inputs have to do. So, having very briefly established the relevance of it, all we're going to do is extract DNA from soil. We're going to snip out what serves as essentially a molecular fingerprint to what organism did this DNA come from. We're able to use that information to figure out the entire community and on that basis tell you here's your soil's capacity to provide certain crop influential, crop benefiting, or crop harming services, producing plant growth hormones, suppressing root feeding insects or nematodes, suppressing disease, mobilizing plant nutrients, immobilizing plant nutrients. This is going to be a quick rundown of what is going to be measured by this test. I will save going into detail for the after you have your results thorough walkthrough, how to use this data. Reach out to me if you've already taken the test and that's what you're looking for. We will assess soil-borne bacterial and fungal pathogens and their risk level benchmarked for your crop. Remember, disease is a function of the presence and abundance of the pathogen, this bottom left corner, what's being measured here on this page, the susceptibility of the crop. We will tell you a little bit about that by telling you about beneficial organisms in the microbiome and a favorable environment. This test has no way of telling you if the environment is currently favorable to this disease, switching into infection, virulence mode, and proliferating and attacking your crop. So you still need scouting, need a fuller picture to predict disease, but this is a great risk measure for making some decisions about variety rotations. I'm already digressing. So we'll also tell you organisms that with slight risk, organisms that were undetected. We'll tell you about biocontrol agents. This is the police force. These are the ones that suppress disease. We've got organisms that suppress fungal pathogens, organisms that suppress bacterial diseases, organisms that can kill or protect crops from insects, and those that can protect crops from root-feeding nematodes. There's a section on nutrient cyclers. Nutrient cycles, your regular soil nutrient test, remember, will look for the inventory. What are the amount of currently available nutrients or readily available nutrients in this system? We're not looking at that. This is a DNA test. You have your soil nutrient test to look at that. We're providing insights into the blind spot, the workforce, those that move the inventory. Take the unavailable nutrients, make them available. Take the available, compete with the crop for them, so on and so forth. Or not necessarily compete with the crop, immobilize them so they're not, in the case of nitrogen, leached from the system. So nitrogen, all the pathways, most of the pathways in the nitrogen cycle are governed by organisms. Your soil nutrient test is going to tell you about nitrates, ammonium. This test is going to tell you about the organisms that can transform nutrients from unavailable forms like organic nitrogen or gaseous nitrogen into a plant available form. I'm not rushing this too much, but I want this to be concise. So for instance, nitrogen pathways, we're going to tell you about nitrogen fixers, take gas, turn it into organic nitrogen. We'll tell you about mineralizers, these ammonifiers and nitrifiers. We've been talking about mineralization for many years. This is a measurement of the organisms that perform that function. We've been using it as a concept, but not measuring it for many years in agronomy. We have on the loss side of the equation, denitrifiers that take organic nitrogen, or they take inorganic nitrogen and they gas it off into the atmosphere. That's lost fertilizer. You have organisms that specialize in breaking down urea. The higher this is, the more important it is to stabilize your nitrogen. And we have immobilization, loss of nitrogen to microbial biomass. That is only temporarily unavailable and is a great way to prevent leaching if your system is prone to leaching. You'd like microorganisms to take it up. Phosphorus, without dwelling on it, we're looking at pathways that unlock unavailable inorganic phosphorus and unavailable organic phosphorus. That 12.6% is the common levels of phosphorus use efficiency, the amount of fertilizer phosphorus applied that gets into plants. So these are the organisms in organic P-solubilization, the solubilizers, the mineralizers, who take unavailable organic and inorganic phosphorus, flip that in the order, and make it available to the plant in the form of phosphate. And then we have the immobilizers, those that are also taking up and utilizing soluble phosphate. And then finally, without spending too much time on it, same deal for potassium. Structural potassium, there are hundreds of years of potassium in most systems. It takes microbes to take that potassium and make it plant available. And then there's our competitors, immobilization. Micronutrients, these are 8 metrics that assess the potential of your soil microbiology to mobilize these nutrients into a plant available form. The ideal score is going to be between medium and high. Okay, carbon, we've got those that take CO2 from the air and fix it into organic carbon. And there are these organisms that break down complex organic matter into simple organic matter, decomposers, residue degraders. On the loss side, this is about oxygen preference. What's our load of oxygen breathers, aerobes? What's our load of anaerobes, those that can get by all right without oxygen? What's our load of methanogens? They cannot get by in the presence of oxygen. They are only active in low to zero oxygen environments. That I'll save it for the results. That informs us about whether your soil is well aerated. And then finally, there are sections on plant growth promoters who produce PGRs, plant growth hormones, and stress hormones, as well as other stress mitigators. So here's our plant hormone pathways. On the growth promotion side, we'll look at organisms that can produce auxin, cytokinin, gibberellin. These govern growth stages. I don't need to belabor it. You all understand PGRs. You understand your crop's stages of growth and the transitions in between them. And then on stress mitigation, we have the glue makers. Exopolysaccharide, don't be intimidated by it. It just means glue. These are chains of sugar outside the cell. They retain nutrients. They create soil aggregation, water-stable aggregates. This is what governs how well your slake test goes. We've got our heavy metal resistant plant growth promoters, our salt resistance plant growth promoters. We have our iron chelating microbes. These produce siderophores. It's an iron chelating molecule. We have, on the hormone side, organisms that reduce ethylene stress by reducing ethylene production in the plant. We have those that produce salicylic acid, which helps with abiotic stress resistance and triggers immunity against various biotrophic pathogens. And then finally, abscisic acid. This is the molecule that helps deal with various abiotic stresses, but especially drought, especially water availability. For instance, it governs when the stomates open and close to control evapotranspiration loss. So think of these organisms as an intelligence network in the soil, informing the plant in real time about changes in water availability. These are 2 plants, exactly the same plants, exactly the same number of days since watering. I think it was 5 days since watering. The only difference between these plants was the inoculation of a stress mitigating Pseudomonas bacterium. The only difference between these 2. So if you've got 2 plants in the exact same conditions, the higher those stress mitigation scores, the more likely your plant is going to be able to push through stress, continue growing, continue photosynthesizing, continue filling grain, and so on. Lastly, we're going to give you an overall, not lastly, almost lastly, we're going to give you an overall network score, a community score. This is basically a look at how well integrated this microbiome is. Is it disjointed? Is it competitive? Is it non-cooperative? Or is it completely well integrated, collaborative. This is a score that will tell you that essentially. It's a full community analysis score of soil biological health. And then there's going to be a total count of bacteria and fungi. If you're comparing between two samples, which I strongly recommend, then this is worth looking at to see, is one soil versus the other, particularly providing a greater environment for microbes, greater fuel for them to act. So I suggest strongly having two samples to compare. That will give you a benchmark. Have your strong yielding, healthy zone. You would like the rest of your farm to operate like this zone, and the poor area, perhaps chronically low yield, perhaps facing some issue that you think may have a biological cause, haven't figured out other causes. Submit those two samples so you have a benchmark for which scores are meaningful. If you request it, or you're sending us a sample through one of our licensed lab partners, there will be a physical chemical component that is also included on the test, as well as through the lab partner's original test. Passing ahead. All that's basically what gets measured. There's a couple free tools online. This is a tool that is going to let you compare two samples or groups of samples, and it'll just highlight the biggest differences between those. If I click on one of these, if I click on salicylic acid, I'll get a box plot where each of these dots is one of the samples, and I can see their spread. I can see what their actual 0 to 100 functional scores are. I can do the same thing for organisms. These are box plots of bacillus across 20 different samples that I've put in 3 groups. You can also, instead of clustering them, have each sample be a line in a heat map so that you can look for common patterns or distinctions between patterns between a group of fields and samples. You can look at the organisms in a given sample, and then finally you can look at the similarities and differences between the microbiological communities of different clusters or groups of samples. So if I may quickly, if you would like to order BeCrop tests, you go to stores.biomakers.com/producto/BeCrop-test. That is a way you can buy 1 to 10 tests. If you want to buy a larger sample package, you'd reach out to me so that we can, basically I'd talk to you, figure out your objectives, and we work out a strategy together that is the best bang for your buck. If you already work with one of our lab partners, Midwest Labs, Waters Labs, or Regen Ag Labs here in the US, you can go to info.biomakers.com/US-lab-partners. And there is information about how to submit samples for DNA testing through any of them. If you want to reach me, I'm Scott McElveen. There's my email address. Thank you for your time. I probably went longer than I meant to. Hopefully that is plenty of information to make a decision about whether this is the test you're looking for. Bless you, sir or madam, your crops, and your critters.